Welcome to ClimTip Webinar series. Today we will be talking about marine life and the effects of Atlantic Ocean tipping elements, specifically North Atlantic Subpolar Gyre and the AMOC, which is very interesting because we haven't covered biodiversity related topics, I think, for a while now. And we're happy to have Amber Boot from Utrecht University giving the talk today. A few words about our guest speaker. Amber started her PhD at Utrecht University in 2019, focusing on the interactions between the marine carbon cycle and the Atlantic Meridional Overturning Circulation, or the AMOC, using both simple box models and comprehensive Earth system models. Amber is currently a postdoctoral researcher in the same group, studying the interplay between the North Atlantic Subpolar Gyre and the AMOC, as well as the modal biases influence AMOC stability, right? How biases influence AMOC stability. The work I will present today stems from a complementary research line exploring how marine ecosystems respond to tipping behavior in both the Subpolar Gyre and the AMOC. And with that, I'll hand it to Amber.
Yeah, thank you so much. Can you hear me all right?
Yes, we can hear you.
Great, then let's get started. Yeah, thank you for that welcome, and thank you for the opportunity to present here. Yeah, so I suppose a lot of you were also at the EDU last week, and I hope you recovered a bit. I haven't really, so but I'm sure it will be all right. And I'm excited to present this work here today. I will be talking, like was already mentioned in the introduction, like about marine ecosystems, and I separated the talk a little bit in three parts. First, I will give a little bit of a marine ecosystem, like 101, a very short introduction to marine ecosystems, so you know what's going to happen in the results section. Then I will talk about a recent publication that we published earlier this year, which really focuses on the AMOC. And then at the end, I will also include a little bit of work that I'm currently working on, so that is still very preliminary, and that focus on the Subpolar Gyre. So I also have my own title slide. Of course, the main reason here is to show off the drawing I made, but no, of course, it's also to because I want to acknowledge my collaborators, because I did not do this research alone. So Emma and Iris are the two PhD students at our institute that I work with now on the new stuff. Jeroen and Marta are help me with the publication with the marine ecology model and the interpretation thereof, and I work together with Anna and Henk during my PhD, which this research started, and well, I'm currently still working with Henk during my postdoc. Yeah, and before I start with the what I call the marine ecosystem 101, I also always like to start with a question, and that is why should we actually even care about marine ecosystems? Because this is also an important question for me for why I do this research. And also one of the things that I find so cool about marine ecosystems is that there are so many different reasons why you can care about these ecosystems. So for example, they sequester carbon. Without marine ecosystems, atmospheric CO2 concentrations could be 200 to 400 ppm higher than they are now. They oxygenate the atmosphere. So on human timescales, around 50% of the oxygen in the atmosphere comes from marine photosynthesis. They provide food. Around 11% of the population on Earth has fish as their main protein source. And well, through fisheries and through tourism, they also, which are multi-billion industries, they also are a very source of income. So these are really typically things we would call like marine ecosystem services. But I think we should also go a little bit a step further away from the services, and we can also have this moral argument that, well, next to us humans, maybe even despite us humans, these marine ecosystems, they simply have a right to exist, and this is also very important to me. Now, what I think is, well, obviously for me, the most important reason is that I think marine animals are just very cute. I mean, look at these guys. And this is my absolute favorite, the penguins. So adorable. But yeah, I mean, all the cute animals aside, I think the point I'm trying to make is clear, right? If it's from an Earth system perspective, a socioeconomic perspective, moral, individualistic, I think we should argue that marine ecosystems matter and marine ecosystems are vital. And these marine ecosystems, they are under threat by anthropogenic activities such as pollution and overfishing, but also by climate change, by global warming. And this will be one of the focus points during this talk. Now, let's talk more about the functioning of marine ecosystems. And I want to start with the food web. So at the bottom of the food web in the ocean, the lowest trophic level we have are phytoplankton. And phytoplankton are photosynthesized, and they are basically, well, the plants, the trees of the ocean. Now, and typically in the ocean, it's not universally true, but small organisms get eaten by larger organisms, get eaten by even larger organisms that get eaten by even larger organisms, right? Now, what I show here now is a nice food chain. But in reality, we do not have a food chain, we have a food web. So still very simple, but I think the idea I try to bring across here is that we have different groups, and each group can have like multiple prey, so multiple food sources, but also multiple predators. And this is still very simple, but I will show a figure later on about the food web use in this study, and that is much more complicated. Now, these food webs, they can be affected in a bottom-up way, and that basically means we change something in the lower levels, and that this effect cascades through the food web to the higher trophic levels, and we can have top-down effects that we change something in the top predators, which influences lower levels. Now, a great example of such a top-down constraint is, for example, fishing. But we will not consider that today. So I will not be talking about fishing, which means we will mostly deal with like these bottom-up effects starting at the phytoplankton level. Now, these food webs, they exist in an environment with a few important variables and boundary conditions. One important variable is temperature. So this has an effect in two ways. So a lot of biological processes depend on temperature, but also each group has its own thermal niche in which they can thrive. So a certain temperature range in which they find comfortable. So temperature very much explains what species can live where in the ocean. Then we also have like nutrient concentrations, and these are important for phytoplankton because they need these nutrients to photosynthesize. And then we have also ocean depth, for example. Just like we have a thermal niche for each group, also each group has a certain depth level where they can live. Right, not all species can live at the surface and at the bottom. There is some difference there. Now, because we will be talking mostly about the effect of phytoplankton cascading upwards into the food web, I have also a slide about more detailed about phytoplankton. So we have an ocean here, and in our ocean we have a surface layer here above the dashed line, and this is around like 100 meters. And then we have a deep ocean. So this drawing is definitely not up to scale, right? And in our ocean, we have dissolved inorganic carbon. We've got some nutrients such as nitrate and phosphate, and we've got our phytoplankton. And as you can see, the phytoplankton are only in the surface ocean. So they are only basically in what we call the euphotic zone, which is basically the region in the ocean where sunlight can penetrate. So this is really confined to the upper layer. Now, and what happens with these phytoplankton is we get some carbon, we get some nutrients, we have a phytoplankton, add some light, we have an increase in phytoplankton biomass, right? This is photosynthesis. It's as simple as that. Except that the way how our ecosystems and the ocean work is that our nutrients are relatively low in the surface ocean and high in the deep ocean, whereas our phytoplankton, they live in the surface ocean. So here it becomes clear that it's very important to have like ocean circulation. We've got to have like some sort of mixing and training nutrients from the deep ocean into the surface ocean to enable basically photosynthesis, to enable production and an increase in biomass in the surface ocean. Yeah, so we will put this talk also in the context of global warming, and there are several ways global warming can affect marine ecosystems. I decided to highlight two, just to give a few examples. And the first one is biogeographical shifts. So what we see here on the left is an ecosystem where we have some sea ice with a penguin on there, and we have a killer whale here in the ocean. They're all just chilling out there. And then what we get ocean warming, global warming, and then the sea ice disappears. This region is unsuitable now for our penguins. So the penguins disappear out of this ecosystem. Now we see the killer whale is still there because killer whales have quite a large thermal niche in which they can thrive. So they don't do a lot. But now we can see that there are new species basically invading this region because now this region has become suitable for them. Well, and here I drew a marine turtle, which is actually very unrealistic that we go from penguins to marine turtles, but I think it brings across the point I'm trying to make. So this changes basically how our ecosystem looks, how our ecosystem functions, and also the ecosystem services they can provide. And I also want to highlight this does not necessarily mean that all what kind of metrics you want to attach to them are negative, right? The main point is that the ecosystems will just look different. And the second point is stronger stratification. If we have global warming, the stratification in the surface ocean might become stronger. And on the previous slide, I explained the importance of the entrainment of nutrients to the surface ocean. And in the stronger stratification, this might become this entrainment might become weaker. So we might see a decrease in productivity in the upper layer. Though this physical mechanism is quite clear, what we see in the climate models, however, in the CMF6 models, that this is not necessarily the only thing that's happening. So in the climate models, it's much more complex than just stronger stratification equals less production. Yeah, and that basically ends I think for now, all you need to know about marine ecosystems for this talk. And we're gonna put these in also in the perspective of our Atlantic Ocean tipping elements. And I suppose we are all familiar with this plot. Some of you might also have used it in your own presentations, just like I did now. But we will be focusing on the Labrador Sea and the Subpolar Gyre collapse and the Atlantic Meridional overturning circulation collapse, or the AMOC collapse. Now, since this is for ClimTip, I assumed that everyone is slightly familiar with these tipping elements, and I will not go into a lot of detail about how these tipping points function, basically. But just to give a little bit more background for those who need it, we can visualize the AMOC as this surface current that flows from the South Atlantic all the way to the North Atlantic, and that it transports a lot of heat. And then here in the North Atlantic, we have deep water formation areas in the Nordic Seas, in the Erminger Sea, in the Labrador Sea, right, which are part of the Subpolar Gyre. And then we have a deep water current flowing southward as a return current. Now, in this study, we see here that it's expected that the global warming threshold for the Subpolar Gyre, the SPG, is smaller than 2 degrees, and for the AMOC, it's larger than 4 degrees. So these are warming levels that could happen under future climate change. And now at least this is what I've been thinking about recently a lot, is a little bit about the semantics about what do we call a collapse and what do we not call a collapse. And I don't want to get into that right now, also because I think it's a much larger discussion. So I will try to be a little bit more specific with what I mean. So if I talk about the Subpolar Gyre, I will talk mainly about the cessation or a strong reduction in deep convection, which have local effects on the climate system and are relatively fast on timescales up to a few decades. And if we talk about the AMOC, I'm talking about a strong reduction in AMOC transport. So this is volume transport, but also heat transport with global effects on the climate system. And they also have this relatively fast timescales, but really also these longer timescales up to like a century, right? And for example, the results I will show, some people will call that an AMOC collapse. Some people will call it an AMOC weakening. And I think at the end of the day, it's a little bit about semantics, especially if you talk about the timescales I'm talking about. So I will mainly talk just about the effects of an AMOC weakening on marine ecosystems is the end goal. So I hope that that's a bit clear, because the end goal basically of the question I try to answer here is what is the effect of the Atlantic Ocean tipping elements on marine ecosystems? So like I said before, I want to focus first on the AMOC, on the publication we already published the paper, and then I will later focus on the Subpolar Gyre. But the end goal is, of course, to know what happens to our little friends we met earlier. Now, the results I'm going to show you are based on Earth system model simulations. And for this, we use the Community Earth System Model 2, CESM2, at a 1 degree resolution. So we have an atmosphere model, we have a land model, sea ice model, and our ocean model. And our ocean model POP2 contains the ocean dynamics, the ocean physics. And then we have MARBLE, and MARBLE contains our marine biogeochemistry. And I will talk a little bit more about it on the next slide. Now, we forced this model with 2 emission scenarios. We have the SSP1 2.6 here in blue and the SSP5 8.5 in orange. And you can clearly see, right, this is quite a big difference in forcing conditions in 2100. Now, in all simulations, they are run from 2015 to 2100. And for all the line plots you will see, I smoothed them with a 5-year moving mean and cut off the first 5 years. So the graphs will start in 2020, but the simulations start in 2015. So all simulations start basically at the same point, but due to the way I plot them, they seem to be already diverging earlier. So that's just a way of plotting. But I will highlight it later if that's might be confusing. Now, for each emission scenario, we have 2 types of simulations. We have a control simulation in which we just use the carbon emissions, and then we also have a hosting simulation where we also have these same carbon emissions, but then we select this region in the North Atlantic and we just dump a lot of fresh water on there at a rate of 0.5 Sverdrup. And well, like several of you probably know, this will result into a strong AMOC weakening. And then we can compare the hosting and the control simulations to see what this additional AMOC weakening actually does to the climate system and marine ecosystems. But before we go to the results, I will talk a little bit about MARBLE, the Marine Biogeochemical Library. And this is based, just as many other biogeochemical models, on what we call an NPZD model, which stands for Nutrient Phytoplankton Zooplankton Detritus model. And in this model, we have 4 different nutrient types. So we have nitrogen, phosphate, silicate, and iron. And together with temperature and light, they co-limit the production of 3 different phytoplankton groups. And then we have diazotrophs. So these are very small phytoplankton, and they form only a very small fraction of the total biomass, the total phytoplankton biomass. So for today, we're going to ignore these little guys. They are also not so important for the results. Then we have diatoms. So these are larger phytoplankton groups that form these silicate shells. So they can be limited by all 4 nutrients we see here. And they typically prefer like more turbulent regions with larger mixed layer depths. And then our third group is the small phytoplankton group. And well, surprise, surprise, they are small, relatively small compared to the diatoms. And they do not construct silicate walls. So they are only limited by nitrogen, phosphorus, and iron. So that's a clear difference between the 2 phytoplankton groups we will discuss today. Now, these phytoplankton groups are grazed upon by 1 zooplankton group that also have higher order grazing parameterized. So this higher order grazing basically is the rest of our ecosystem, and that is a parameterization. So our ecosystem here in CESM2 consists of basically 3 phytoplankton groups and 1 zooplankton. Okay, that's all I had to say about the model and the experiments. So now we can get to our results. And we start with the AMOC weakening we simulate. So this is the AMOC strength at 26.5 degrees north. And in all the line plots I will be showing, the solid lines are for the high emission scenario, so the 8.5 scenario, and the dashed lines are for the low emission scenario. The blue lines are for the control simulations, and the orange lines are for the housing simulations. I mean, the legend will be next to every plot, but just so you know that this is consistent over the entire presentation. Now, in blue, in the control simulations, we see that the AMOC already has quite a large weakening, right, of around 50%, just due to the greenhouse gas emissions. But we also see, right, this in orange, we see this clear force response to the housing. We see a much stronger and faster weakening compared to the control simulations. And if you look at the difference between the housing and the control simulations, right, this is also what we clearly see. We see this sort of what I would call a skewed like V shape with a minimum around the 2040s, 2050s, and then the difference between the simulations decreases again. And we will analyze the AMOC weakening response basically by taking the difference between the housing simulation and the control simulations. Now, both the forcing, the climate forcing, the greenhouse gas forcing, and the AMOC weakening cause the response in global mean surface temperature. So that is what I show here. Well, what of course stands out is this very clear difference between the emission scenarios, where the high emission scenario leads to a warming of around 5 degrees over the century, and the low emission scenario of about 1 degree. And also in global mean surface temperature, we see this skewed V shape, right? This is this clear AMOC weakening signal that global temperature cools due to the AMOC weakening. Now, it's not just a cooling on a global average, but there's also this spatial pattern. So this is for sea surface temperature. And if I show these kind of figures, what you will see here is that it's the housing simulation averaged over the last 5 years minus the control simulation averaged over the last 5 years of the century. Right, and here we see sea surface temperature, and we see this clear regional pattern we expect from an AMOC weakening, where we see this clear cooling in the northern hemisphere and warming in the southern hemisphere. So this is for the low emission scenario, and this is for the high emission scenario. And we see these very similar patterns, though the amplitude is a bit smaller here in the high emission scenario. And we have this region here in the North Atlantic. And that is caused by that in the high emission scenario in the housing simulation, the North Atlantic current shifts a bit northwards. So we get warming on that side compared to the control simulation. Now, we get also some responses in the atmosphere. So we get changes in precipitation, for example. So this is also this southward shift of the intertropical convergence zone, which we expect following an AMOC weakening. We get changes in the zonal wind stress, for example, with an increase in the trade winds here, and a southward shift of the Southern Ocean westerlies. And basically what the changes in temperature, in the precipitation, in the wind stress, they change the vertical structure of the upper ocean. They change stratification, they change mixed layer depth. And this has an effect on our biogeochemistry, which I will talk about later. And then the temperature effect also influenced the Arctic sea ice extent, Arctic sea ice fraction. So this is sea ice fraction. And here on the left is the low emission scenario. And you see that in the housing simulation, we've got much more sea ice here in the Barents Sea and around Iceland and around the tip of Greenland. And in the high emission scenario, we see mostly increases here in this region and also around the Canadian archipelago. And here you can also see the clear difference between the two emission scenarios, which is maybe a little bit clearer on this figure. So this is the same figure again. And here, well, the contouring in the background is not important. That is sea surface temperature, but that's not relevant for what I'm trying to show here, because I want to focus on the lines here. And this is the 0.15 sea ice fraction line as average over the last 5 years, where the dashed line is the control simulation and the solid line is the hosing simulation. And you can clearly see basically what we see also in the figure over here, this strong increase in sea ice. The sea ice edge moves southward here, and we see much more sea ice in this region. And you can also see that in the high emission scenario, there's just much less sea ice overall, where the sea ice edge really retreated more towards the Arctic. And this is also what we see later on in the ecosystem response, is that qualitatively, the low and the high emission scenario behave very, very similar, except in the Arctic. And it is mostly this region where we will see a different response. Yeah, and so like I already said, all these changes in the physical climate system also affect our biogeochemistry. So this is for the low emission scenario, nitrate concentrations integrated over the top 150 meters of the water column. And there I do want to note is that nitrate is not necessarily the limiting nutrient everywhere in the ocean. So we have 4 different nutrients, and nitrate is just one of them. But this is just to give an indication what happens to nitrate. And these are the initial distributions of nitrate. And here you see the effect of an AMOC weakening. So there are regions with quite a big decrease. For example, here. And these are of the order of around 50%. So this is really, really large decreases. And here we see, for example, a large increase. But what I think really stands out here as well is we have this really regional response. This is really a patchwork of regions where we see an increase and regions where we see a decrease. And we see the same in the productivity of our ecosystem. So this is net primary productivity. And we see that mostly in the Atlantic Ocean, in both emission scenarios, we see these large decreases in productivity, whereas in the equatorial Pacific, we see these increases. And again, it's quite similar what we see in the different emission scenarios. And now to get to basically where we want to go to what happens to the biomass in our ecosystems. And that is for now the phytoplankton biomass. And I will show them at first only for the low emission scenario. So here in the top left, you see the original distribution at the beginning of the simulation of small phytoplankton. And here that is for diatoms. And you can clearly see that basically where there are small phytoplankton, there are no diatoms. And where there are diatoms, there are no small phytoplankton. That's a little bit the idea. They do not necessarily like to coexist that much. And this is also what we see in the response to an AMOC weakening, where we have regions, for example, here in the extension of the Gulf Stream, where we see an increase in small phytoplankton, but we see a decrease in diatoms. And here we see an increase in diatoms, but a decrease in small phytoplankton, right? So this is, they balance a bit, but this in the end does change total phytoplankton biomass. And that is an important variable. So that is all phytoplankton groups summed together, and then a response to an AMOC weakening. And we get I would say, the same patchy response of increases, for example, here in the North Pacific and the Equatorial Pacific, but we also see these decreases, strong decreases in the Atlantic, here in the Arctic, which, by the way, like I said earlier, is not the case in the high emission scenario. So this is the main difference there. Now, globally integrated, we do see a decrease in total phytoplankton biomass by a few percent. But what clearly stands out is this is really clear regional response. Okay, well, now we know what happens to the total phytoplankton in our marine ecosystems. But like I already showed in some of my drawings in the introduction, marine ecosystems are much more than just phytoplankton. Phytoplankton are just the base of the ecosystems. So for me, this was not sufficient. I wanted to know more. I wanted to know what happened to the entire food web. So what we did is we also used EcoOcean. And EcoOcean is a marine ecosystem model. There are several different marine ecosystem models. If you're interested, you can check out, for example, the FishMIP papers, where they compare some of the marine ecosystem models. And EcoOcean is a model that simulates 52 functional groups that represent more than 3,400 species. So to compare it to CESM, where we had 4 functional groups, right? 3 phytoplankton groups and 1 zooplankton group, right? We have way more groups now. And basically how these groups are made is they look at certain species that have similar traits, and then they just group them together so that you have groups with the same ecological traits, basically. Now, a few examples of these groups are different size classes of phyto and zooplankton. So we have 3 phytoplankton groups, so the same that are in CESM2, and now we have 3 zooplankton groups. We have also many fish groups, and they are, for example, differentiated in size. So we often have a small, medium, and large group, but also where in the water column they live, and also some other qualities. They're also differentiated on that. We've got a couple of mammal groups, such as dolphins and porpoises, toothed whales, baleen whales, and there are also some other groups, some crustacean groups. So it's quite a complex model with complex food web dynamics and also some habitat modeling. And with that habitat modeling, basically for each grid cell, the model looks at what is the temperature, how many prey are there, how many predators are there, and it decides a little bit how suitable is a certain grid cell for a certain group. And we force this model offline by phytoplankton and temperature output from our community Earth system model 2 results. And then we can construct a figure like this. So this is total consumer biomass, and that is all biomass on trophic levels higher than the phytoplankton groups. And well, one thing that really stands out immediately is that the high latitudes seem pretty messed up. And there are 2 reasons for that. One, this ecosystem model is run on a rectilinear grid. Not my choice, but yeah, that's what I had to deal with. And it's also because of sea ice, and this causes quite a bit of internal variability in certain species. So when I analyze these high latitude regions, I make sure to smooth them over in time and to take larger regions to integrate them over. But what outside the high latitudes, so the lower latitudes, we see again a very similar figure as we saw, similar pattern as we saw for the total phytoplankton biomass with these regions where we see increases, but also where we see decreases. And it becomes especially apparent when you put them next to each other. So this is only for the low emission scenario. This is the ecosystem model output, and this is the Earth system model output, and they are quite similar. Now, based on these figures, we decided to do a couple of regional case studies to figure out now what's happening here actually. And I will discuss 2 of them today. So I will look at the subpolar gyre, but first I will look at the subtropical gyre. And that is actually quite a bit of a boring one, but it shows clearly what's happening. So I will now look at the variables integrated over this, averaged over this region. And here I show small phytoplankton and diatom biomass. And what we see in the control simulation, so the blue lines, that small phytoplankton biomass shows a moderate increase, while diatom biomass shows quite a large decrease. And in the housing simulations, we see this very sharp, strong decrease in the diatoms, which is very similar actually what we saw to the AMOC weakening. And as the diatoms decrease sharply, we see first a sharp increase in small phytoplankton biomass, followed by a leveling off and a moderate decrease in a low emission scenario and a strong decrease in the high emission scenario. Now, what really drives important driver in this region is the change in nutrient limitation in diatoms. So basically what we get is that the concentration of nutrients decreases in this region. And this causes the main response we see here in the diatoms. So this is the dominant thing that's happening here. And what we see that as the diatoms decrease, basically the small phytoplankton group can profit from basically the space that's left over from the diatoms. Now, if you look at total phytoplankton biomass in CESM2, we see that it really follows the response in the diatoms, right? We see this decrease here. And if you look at the EcoOcean response, right, this is very similar. Basically, what happens in CESM2 also happens in EcoOcean. So here in this region, this ecosystem model doesn't really have a lot of added value, I would say. But then we go to the subpolar region, and there interesting things happen. So this is, again, small phytoplankton biomass and diatom biomass. And what we see in the control simulation is this very strong decrease in diatoms spread out over the entire century, basically. And this is accompanied by also an increase in small phytoplankton biomass. But this increase is smaller than the decrease we see here. But what we see at the end is that in the control simulation, at first we have diatoms that are dominant, but in the end we have small phytoplankton that are dominant. So we see a shift in phytoplankton dominance. Now, if you look at the housing simulations, this initial decrease in diatoms, so the diatoms here in the housing simulation also start around here, is much sharper and then levels off. And the small phytoplankton see a sharp increase and then level off. Now, this initial decrease in diatoms, this is again related to changes in the nutrient limitation. So we see this reduction in nutrients there, and that causes the production of diatoms to decrease. Now, just like in the subtropical region, when we get a decrease in diatoms, the diet limitation for small phytoplankton is lifted. So they are able to profit from basically the fewer diatoms in the ocean, and they are able to outcompete the diatoms. Now, what we see here in the small phytoplankton, so in the control simulation, we see this decrease that continues the entire century, and here we see this leveling off in the housing simulation, and this is also related to very strong temperature limitation in the housing simulations following the AMOC weakening. So we get a very strong cooling, and that limits also the productivity in this region here. Now, if you look at total phytoplankton biomass, we see this really strong decrease of more than 50%, of around 50%, and we see that actually the AMOC weakening, the housing and the control simulations are around the same point at the end of the simulation in ESM, in the CESM2. But this is not the case in EcoOcean, because what we see in EcoOcean is, well, total consumer biomass in the housing simulations decreases. It more or less follows what we see here in the orange line, not completely, but right, the approximate trend is there. But if you look at the control simulations, where we get around 50% decrease in total phytoplankton biomass, we get a 10% increase in total consumer biomass. So this clearly shows that the higher trophic levels in our ecosystem respond completely different to the forcing than the total phytoplankton biomass. And this is something interesting, which I will talk about later, which we did not really discuss in the published paper. Now, based on all these case studies and all the analysis we did in the paper, we tried to come up with a mechanism of what's actually happening here. And I will go over that quickly. So in our simulations, we start off with an AMOC weakening. And what this AMOC weakening does, it disrupts the northward tracer transport. And the disrupted northward heat transport results into a response in the atmosphere, for example, the precipitation, but also the zonal wind stress, and also in our sea surface temperature. And this changes the vertical structure in our ocean, which affects the light availability and the three-dimensional nutrient distribution, which in turn is also affected by the disrupted nutrient transport. Now, both the temperature, light, and nutrient response influence what happens to our phytoplankton groups. And it really depends on the region and which response is dominant. This is also what makes this so incredibly complex and difficult to figure out what's happening. Because these are all nonlinear responses, basically. And then this all happens in our ESM. And now, if in our ESM we do not get a phytoplankton composition shift, such as in the subtropical region, right? It's quite boring. Our total consumer biomass just simply follows our phytoplankton biomass. But if we do get a composition shift, we get a deviating response, such as we saw in the Subpolar Gyre. And this could be a more positive response, but it could actually also be a worse response. So in the Subpolar Gyre, I would say we saw a positive response, but for example, in the extension of the Gulf Stream, we see a negative response, a worse response. Now, I've remained quite qualitative so far, but I also want to give you a few numbers. So what we see globally integrated is that marine ecosystem biomass decreases by around 4% due to the AMOC weakening, which is on top of a 2% decrease due to climate change. And this all happens in the low emission scenario. And in the high emission scenario, these numbers are 2 and 12%, where the change in the 2% is in large part what happens in the Arctic Ocean. But this is globally integrated. If you look at total consumer biomass on a local level, we see that decreases can be as large as 30% due to the AMOC weakening. And if you go for specific groups in the model, this can be even larger. So I would argue that an AMOC weakening is an additional threat to marine ecosystems, and therefore also to socioeconomic systems that depend on biodiversity for food and income. And that is basically what we try to write down in our publication. And now I also want to spend like 5 minutes talking about the Subpolar Gyre stuff I'm currently working on. So this is still very preliminary, and I hope it makes a bit sense, but don't expect it to be all true. What I'm telling you, these are is based on hypothesis, but I wanted to share it anyway. And I want to focus on the control simulations. So there are two interesting things happening in the control simulations. So first of all, it's this phytoplankton composition shift that I was talking about, right? So now I'm talking about the blue lines here. And that we see a deviating response in the food web, where the higher trophic levels respond qualitatively different from the total phytoplankton biomass. And so this is one thing that's interesting there. And the other thing is that, I do not have the CESM2 data is still on archive. I hope to have them available already, so I could not check it. But this strong temperature response here in the 2040s seems to be indicative of changes in deep convection. And we can also see that in the CMIP6 NCAR CESM2 simulations. So this is for location in the Erminger Sea, where in blue you have the mixed layer depth, And then also here in the 2040s, 2050s, we see this decrease in mixed layer depth. And I'm pretty convinced that this is the same signal, basically what we see here. And basically the questions I want to look at here is basically what is the food web response to a phytoplankton composition shift? And how does this relate to changes in deep convection that happen, I think, at the same time? Now, if you look at the food web at the individual groups, you can divide them in 3 different categories, basically in response. So you have a minimum in the 2040s. So this is, for example, microzooplankton. And here you see the blue lines. Here it is minimum in the 2040s and then an increase. And this coincides with the temperature response we saw on the previous slide. Then we have groups that have a minimum in the 2060s, and we have groups that decrease basically over the entire simulation. Now, these categories are not very rigid. I mean, they also flow a little bit into each other, but most groups are quite easy to identify, like in which category they belong. And then I will look into the food web. So this is a representation of the food web. So that is extremely complex. It's really difficult to make any sense out of it. So I'm trying to make sense out of it. For now, based on these connections, I try to understand the response. And I only look basically at the thickness of these connections, but I still need to do a biomass weighted analysis. And I also need to look at the effects of temperature changes. But what my hypotheses are is, well, what I show here. So this is a very difficult, maybe to understand figure, and I don't want you to understand it completely. But what I try to show here is that the gray regions are where are the groups that are decreasing. The blue ones are that have a minimum in the 2060s, and the green ones that have a minimum in the 2040s. Then we have a few that are a bit different, that do not really fall into the categories. The thick outlines are CESM2 output. So these are our phytoplankton groups. And the red outlines are where I'm not really sure what causes the trend. And this is, I would say I'm 90% certain that this is what's actually happening in the model. So not completely yet. And basically what this suggests is that, for example, we see in 48 that has a minimum in 2040s, and this causes the same pattern, the same trend we see in 47, right? So I'm quite certain that these connections are right. And there are way more connections. So this is even more. I'm not certain that this is actually happening. So these connections are also in the model, but they I would not expect them to be very strong, but they can explain the response we see in the groups. And basically what this suggests is that here, group number 40, which are the diatoms, causes the decrease in basically all these gray groups here and the gray groups at this side. And the change in small phytoplankton can explain basically all the green groups here. And then the combined response of 39, 47, and the diatoms explains what happens in 36 and 35, which is then important for these groups, right? So this is which I need to test this with biomass weighting and do a lot more work on it, but this is at least what's coming up. Yeah, so the main part of this talk was based on our publication in Earth's Future. I have a QR code of this publication on the next slide. Other relevant publications are 2 chapters from my PhD, so a publication in GRL and a publication in climate dynamics. With here we look at the phytoplankton composition shift, and here we look at the carbon cycle response in the CESM2 simulations. And I'm currently working on 2, maybe 3, which these are the working titles. So the first one is what I just showed you now. And I also want to look at the winners and losers in the marine food web in response to a strong AMOC weakening, where I will more or less look at the individual groups instead of just at total consumer biomass. And with that, I will leave my conclusion slide up and the QR code of my paper. And then if you have any questions, I'm happy to answer them.
Thank you very much, Amber, for this very interesting presentation. We have a few minutes to, if people have questions. I was, definitely confused about all these different intricate complexities. We have a question in the chat, but also you can unmute yourself and ask or raise your hand if you'd like to ask a question. So Yichul asks, I'm not sure if you would like to read that yourself or I should do that, Yichul. So yeah, on slide 15, you said that global mean temperature response is simply from the AMOC weakening, which is not intuitive for him, he says. As the AMOC weakening is redistribution, hence it is not easy to modify significant global mean temperature changes unless strong regional feedback is accompanied. So that's one question. And the second one, I'm not sure if this is related directly. So here you only compare 5-year mean response, which may be a bit short to accept as a climatological response due to the internal variability. Do you have an ensemble average, he asks, or could you confirm that your analysis is consistent with different time periods averaging?
Yeah, so the time that I only use 5 years, that's indeed maybe a bit short. And it's actually also quite relevant, I think. So I also think like maybe in future publications, I will not do the 5 years anymore. Especially because, well, here you see that this is also the difference between the simulations. There is a very nonlinear trend in the difference between them. So that's definitely one thing I will reconsider for the newer publications to take that time period a bit longer and also look more on the time-dependent response. So yeah, that is one point, and the other one was about the response we saw here. This global cooling. And I have to be honest here that I do not have a good answer for that for you at the moment. So but I can get back to you later on that. But at the moment, no, I don't have an answer.
Thank you. And please go ahead. Richard Wood has a question.
Yeah, thanks. Hi, Amber. Thanks very much. That was a really interesting talk. I've got a question about the ecosystem dynamics. So I don't know much about ecosystem models, but I know that potentially you can get some quite interesting and sort of potentially chaotic dynamical behavior with those models. Your very last slide, I think you were kind of basically you were showing that a lot of the changes in the ecosystem could be just sort of traced back to the bottom level of the food chain, true food web, which is a more deterministic response. So do you have any feel for whether you could get some interesting and variable behavior just from the internal ecosystem dynamics?
I am not sure whether this model is that suitable or that. I know that if you go, for example, the more simple NPZD models, there you can get a lot of interesting dynamics in there. And I'm not really sure if they are necessary in this big ecosystem model. Yeah, and I've tried also. Yeah, I mostly focus on now the bottom-up approach, because I also tried looking more top-down, and that didn't really seem to. That didn't really seem to explain the changes I saw in the groups.
Yeah. Okay. Yeah. So thanks very much. It's great. Really interesting stuff.
Maya has a question. Maya Benyami.
Yeah, thank you for the really interesting talk. I'd read your paper before, and this added a lot more detail. I was wondering at the end when you had your sort of flow chart and you had sort of two different I think you said something about how you can have the sort of shift when you have the differences between the two models, between just phytoplankton, yeah, and total consumer bias, right? So it's the sort of composition shift where you get the deviation, and you said that this deviation can be both bad and good, and you think that in the subpolar gyre it's a good response in some way. Do you have like more details about that? What makes it a good or a bad response, this deviation?
No, this is also something that's still on my to-do list, because there are multiple regions in the ocean where we see like this shift from diatoms to small phytoplankton. And yeah, it's just it's very complex. There's so much output also to look at in the system models. So I do not have a feel for like why in the subpolar gyre we see this response, and in a different region we will see another response. But I also hope that what I'm working on now with really looking into the food web, that will help clear that up.
I guess maybe a more sort of general version of my question is just what makes a good deviation versus a bad deviation, just in terms of the ecosystem.
Then I would say here you see that total phytoplankton biomass decreases. Well, this is right in the, if you look at the blue dashed line, this is, well, almost 50%. And here we see the total consumer biomass in the end increases by around 10%. So here I would say that even though we get a strong reduction in the lowest trophic levels, we get a overall positive response, more biomass in our ecosystem. So I would argue that that's, in a sense, at least in 2100, it's a positive response. But you can also get that where you have, for example, a decrease of, let's say, 20% in total phytoplankton biomass, that leads to a decrease in, let's say, 60% in total consumer biomass. So there you see that even though the change in total phytoplankton biomass is relatively small, the entire ecosystem loses much more biomass.
Okay, cool. Makes sense. Thanks.
Sebastian, please go ahead. We have another question.
Yeah, thank you. I also liked your presentation, and I was wondering about slide 22, I think, where you show the NPP response. Yeah, so this is, yes, the circulation in the ocean is asymmetric around the equator, and the response from a AMOC reduction in the climate is also asymmetric, but here the NPP response looks rather symmetric, maybe not around the equator, but somehow there is this symmetric pattern, right? I was wondering why it looks that way.
So this is also partially related, I think, to the time question I got earlier. So what we see is that initially we see this decrease here is very much related to reduced advection of nutrients due to a weaker Gulf Stream, but also due to what I would call leakage of nutrient poor waters from the subpolar gyre into the interior here in the subtropical gyre. And we see that this decrease really only starts to happen in the 2050s. So there is still quite some asymmetry there, but this is just, I think it's just the time it takes from basically the signal from a reduction in, well, the reduction in the AMOC and reduction in deep convection here, the time it takes to travel to the South Atlantic. And I think those are the main changes we see there. Yeah, does it answer your question?
Yeah, cool, thanks. Yeah, so it really depends on the timing also.
Yeah.
What you see, yeah. Thanks.
Thanks so much. We are over time, but I also was super curious to ask a few questions, because some of the graphs, I don't know if you have time, Amber, to cover that.
Yeah, I have time. Yeah.
Okay. I was curious, that there was this total phytoplankton changes in low emission scenario versus high emission scenario, and then in low emission scenario, which is still global warming, right? There is an increase in phytoplankton, which I found kind of counterintuitive. So I don't know how you could explain that.
Do you mean this figure, or
there was a kind of there was a graph that it was going up. Total phytoplankton.
those? I don't have a graphic increase in total phytoplankton biomass, I think.
Wasn't it? It would go up and then down, something like that.
Do you mean this one, maybe?
Maybe, yeah. Right. Yeah, here it was going down. I think it had another graph next to it, but maybe you could explain based on this one.
Yeah. So what I also tried to explain in the flowchart I showed earlier is that there are temperature plays a role, light availability plays a role, and nutrient availability plays a role. And those processes are not necessarily connected. And this is also there. Then also the region plays a role. Where are we in the ocean? So what we see, for example, here for small phytoplankton, we see here this first initial increase, and that is mainly because we see this decrease in diatoms. So as the diatoms disappear, small phytoplankton basically get more room to grow, and initially they can profit from that. And then these diatoms, they decrease because we have a reduction in nutrients in this region. And there you get this interplay, because small phytoplankton also see this reduction in nutrients. They also feel that they are in the same ocean. But the growth rate of phytoplankton is a function of, well, the amount of nutrients and the amount of light available. And here, the small phytoplankton, they get more light available when diatoms disappear. And this is why initially they get to profit. But you see this especially in the high emission scenario. This sharp decrease here is at some point they also start to feel the nutrient limitation, and they cannot keep up the growth rate they have here. So yeah, I hope that explains a bit what's happening there.
Yeah, thank you. And what about the relationship between the phytoplankton increase, but you mentioned that total consumer biomass is decreasing, and phytoplanktons are producers, right? As I understand.
Yeah. Because the phytoplanktons are primary producers in the ocean, and then total consumer biomass is an aggregated group in our ecosystem model that contains all biomass except basically our producers. So these consumers, they feed on the phytoplankton, and yeah, then the food web does its thing, the temperature does its thing. That's the idea.
Right, and that other graphs, like two graphs that you had, phytoplankton versus increasing total consumer, decreasing total consumer and increasing, or something. All right, like that. This also kind of counterintuitive to me.
Yeah, this is also counterintuitive to me, and this is also why I want to work on this, because this to me is very interesting, but I don't have an answer yet on why this is happening.
Yeah, thank you so much, Amber, and thanks everyone for participating.